This is a plant. Plants are amazing because they have an ability that most animals don't. If we remove a piece, let's just say a chunk of leaf tissue, and we put it on the right growth media with the right hormones, we can grow it into an entirely new plant with very little effort. And it'll be identical in every way that matters to the original plant. In other words, a clone. As such, it'll have the same genes, same growth habit, same delicious fruit, or that same staggeringly high caffeine content. Some plants don't even need special growth media. Just lop a branch off and stick that bugger in the ground. Or maybe water first and then the ground. On the other hand, as you know, if you tried that with a human, at most you'll end up with a few cells that you can keep alive, and probably some legal trouble when people find out where you got that thumb from. And the old stick a piece in the ground method definitely doesn't work, and is even more likely to attract attention, one way or the other. However, that's not to say that cloning animals is impossible, it's just way harder. And while it may be a staple of science fiction, it is anything but. These days, you can pretty casually have your pets cloned, and there are multiple companies that offer it as an everyday service, albeit a crazy expensive one. But what if a world leader wanted to pull a Cleon from the Foundation series, and make younger copies of themselves to rule when they die? Or what if a company wanted to make the ideal boy band, or maybe just a fleet of identical soldiers? Could we clone a person with today's technology, and will the clones even be anything like the original? Today we're going to take a deep dive into this strange world and learn how cloning really works. We'll learn a little bit about the history and where the tech is headed, and then finally talk about how we could give it a try ourselves. Though, speaking of Cleon, have you ever wanted to see what it would be like to live in the Foundation series? Fight space battles, explore different planets, and conquer the galaxy? Well, now you can with the sponsor of this video, Foundation Galactic Frontier, the new game that puts the world of Isaac Asimov right in your hand. It's available on Android, iOS, and PC, so you can play no matter your system. Customize your ship and build a fleet. Colonize and occupy planets, explore the galaxy, and show the universe who's boss. Play alone or with friends, set up guilds, and grow your control of the galaxy until you're a force to be reckoned with. If you want to give it a try, then click the link below and use the coupon code thoughtemporium to get exclusive in-game rewards. Now, while you dream of your shiny new space fleet, let's learn about the strange world of clones. To get started, what makes plants so special that they're easy to clone, and why are animal cells so resistant to the process? Well, the answer of course comes down to the wet clockwork that makes them function. Plants exist in a growth cycle where they just need to execute the same few instructions over and over again until they die. And any part of the plant may need to execute part of those growth instructions. Whereas animals do all of their growing and shaping up front, and then once fully developed only let the cells grow and divide just enough to maintain their function. Here's a really simple example so you understand what I mean. Since plants don't have nerves, they do all of their cross-body communication with chemicals, or more specifically, chemical gradients. The example that we're going to look at is how a plant knows when to make a new leaf instead of just extending the end of a branch. Each leaf on a branch can be thought of as a transmitter, exuding a chemical that, importantly, can't travel very far. As the end of a branch grows, eventually the level of that chemical being detected by the tip of the branch drops. And when this happens, a gene flips on and the make new leaf part of the program runs. Rinse and repeat for the rest of the branch. And the whole plant is like this, be it roots, shoots, flowers, or fruit. Each clump of cells is just listening for chemical signals, like damp radio transmissions, which is also the name of my new punk band. Because plants can't move and have to just react to whatever the environment does to them, at any time, some pocket of cells might need to grow into something else. Maybe some root gets yanked out of the ground, so it'll start behaving like stem. Or a bit gets buried, so it'll start growing new roots there. There's no true top or bottom, there's just plants vibing to chemicals, gravity, and light. Many plant deformities, like witch's broom, are due to overproduction of one of the hormones, and it causes the plants to behave in a very broken way, sprouting enough tendrily branches to make Cthulhu blush. This is why you sometimes need to dip a cutting into rooting hormone, so that the plant knows that that part is supposed to be roots. But there is no equivalent in animals. There's no arm growing hormone, or a bone growing hormone that you just rub on your... Wait, no. Anyway, animals solve the problem of existence very differently. Chemical gradients are nice, but they're also very slow. No plant can pick itself up and take a leisurely hike through downtown to see what human life is like for a change. At least not yet. Maybe in a future video. With animals come both a need for quick, decisive movements, and thus a similar need for a much broader variety of cell types, organs, and tissue specificities. You need muscles to move you, nerves to control them, a brain to decide where to go, and a way to process what you find into nutrients that let you keep going. And since you will need to remove environmental toxins and fend off everything else that thinks you're a snack, throw in a bunch more cell types for that too. For most animals, this is implemented by initially starting with pluripotent cells and then slowly differentiating them into more and more specialized cells, with equally specialized functions. And by maintaining a basal population of stem cells that regularly replace the dying differentiated ones, you can keep the whole system working long enough to find food, a mate, and a comfortable place to die once you've secured the next generation. In nature, most injuries to animals are either minor enough to be healed quickly or rapidly fatal. It's very rare that an animal will have a limb removed or a large hole made in them and then survive. As such, there's not really a reason to maintain pluripotency in every cell. You're probably not going to need to convert your eyeballs into noses. Or if you fall over into a particularly nutritious bit of dirt, you're not going to decide that you may as well start growing some new roots out of your butt while you're down there. Bone normally just needs to be maintained, not constantly grown, shed, and then regrown every time the seasons change. Though, of course, with a few notable exceptions. And thank the fates for that, because if we all became boneless blob men when the days got short, winter would be even more miserable. Now, of course, there are animals with remarkable healing ability, some that are even clonable by just breaking them in half, much like a plant. Split a starfish down the middle and you get 2 new starfish, and possibly a lawsuit from each half's lawyer. And you can press a sea sponge through a garlic press and get hundreds of new sponges. There are also some animals that are called parthenogenic. Basically, these are species made entirely of female members. Rather than mating, they just lay eggs containing clones of themselves, with each generation being nearly identical to the last. Entire populations of nothing but clones. And there are instances of this in animals that don't normally do it as a very rare glitch in the reproductive cycle. It's exceedingly rare, and the likelihood varies by species. For example, recently a group of all-female Komodo dragons that were being kept in captivity suddenly laid some eggs which hatched, but there were no males anywhere to be found.
You're implying that a group composed entirely of female animals will breed?
No, I'm simply saying that life finds a way.
Anyway, if we wanted to clone, say, a chicken, we can't rely on this process. We need to be able to take some tissue, culture it, and then convince it to grow into a new body. Now, growing animal cells on their own is something we've gotten very good at on this channel. We've grown kidney cells on grape tissue, we've grown skin cells on spinach, and of course we regularly grow brain tissue for our neuron project. And for our ongoing meat robot, we'll need to culture both muscle and bone. But even though we can grow all these different kinds of cells in a flask, doesn't mean that we can just turn them back into the body they came from. Part of differentiation is disabling all of the machinery that would allow a cell to regress. As such, the cells will grow and divide, but muscle will stay muscle, skin will stay skin, and so on. So to make a clone, we really have 2 options. We either need to do something to the cells such that they revert first back to stem cells, and then are slowly coaxed in becoming a new embryo. Or we skip that and modify an embryo that's already started. The latter was invented first, and that means it's finally time we talk about Dolly. Dolly is possibly the most famous sheep of all time, which, for sheep, that's saying something. They're not normally the kind to chase fame. Dolly was not the first clone, but she was the first cloned mammal. She was made by the Roslin Institute in Scotland by a process called somatic cell nuclear transfer in 1996. The way this works is pretty straightforward. They start with an egg cell from a donor. This doesn't need to come from the exact animal you want to clone, just the same species. Sperm and egg, as you probably know, each carry half of the genetic code of the parents. When they come together, the halves from each parent pair up, and only then will the egg have all it needs to divide and begin embryo formation. But an egg that's just waiting for a friendly sperm to show up can't. All the mechanisms are there, but the genetic machinery prevents the process from starting. All that genetic machinery, though, is stored in the nucleus of the egg. What if we replace the nucleus in a waiting egg with one from an adult cell taken from our donor animal? Suddenly, the cells will have all of its genetic instructions. All it needs is a push to get it started and something to grow in. Sure enough, that's exactly how Dolly was made. In terms of the hardware, you need a microscope, of course, and the special tiny glass needles. But you also need a way to precisely move the needle around and control the fluid inside of it. This is usually accomplished with a tiny air piston and a micrometer-controlled XYZ stage. For us, these certainly wouldn't be impossible to build, but if you represent a company that makes motorized XYZ stages or a microneedle system, do send us an email and we'd be happy to collaborate. Anyway, once the deed is done and you've made the switch, the next step is to activate the egg. Much like a slinky at the top of the stairs, it needs a little push to get going. And this is going to sound fake, but the most common way to do that is to shock the egg with electricity, which I find gives intense Frankenstein vibes. By using a pulse of electricity, or the application of specific salts, usually calcium or strontium chloride, you disturb the electrical properties of the cell's membrane, and this mimics the natural signals that an egg experiences, which kicks off the division process. This is allowed to grow briefly before it's eventually transferred to a host. As a quick aside, one of my favorite sets of clones are the Mauler twins from Invincible, and in the scene where we first see their cloning process, I was surprised to see that they animated basically what I just described. Mess with a nucleus and zap it to get it to divide. But unlike the Maulers, our current cloning technology uses healthy surrogates rather than a clone tank. For Dolly, and essentially all modern cloning, this means that we need an actual adult female of the species of interest to make a clone, and that individual will give birth to the produced clone. Dolly lived for 6.5 years and gave birth to 6 healthy lambs over her lifetime, so in all respects, she was a pretty normal sheep. These days, as I said in the beginning, the process has been refined quite a bit. Simply by providing a company with a biopsy and a blood sample from your beloved pet, they'll make you a copy. So already an orphan black situation is entirely possible. If somebody wanted a clone and they could convince somebody to make one for them for the right price and coordinate a surrogate, it could be done. And for all we know, somebody already has a freezer full of clones on an island somewhere. And this technology is only marginally more complicated than modern in vitro fertilization. As it stands, people that go the IVF route get a huge selection of options about their newborn. For example, did you know that it's apparently very easy to select for only male or female embryos? I would have thought that it was hard, but no. Male sperm are just lighter and female sperm are heavier. As a super fast refresher, in the most basic case, a male will have one X and one Y chromosome, while females will have two X. There are a lot of exceptions to this, but that's the basic idea. Anyway, the Y chromosome is physically a lot smaller than the X chromosome. There's less code, and with DNA, code length directly equates to mass. So if you separate sperm by weight using a centrifuge, the heaviest, and therefore female sperm, will end up on the bottom, and the lighter males on top. Anyway, tangent aside, the point is that on the lab side, all of this handling is very much just standard practice, and cloning is barely an extra step for a lab already set up to do this. Though there is another problem with this. Much like in an Orphan Black situation, each clone is going to be the same only in appearance. Brains are a lot like the coral that they resemble, in that even two genetically identical bits of coral will grow a little bit differently every time, and will never be a perfect copy. So clones, much like many identical twins, will have wildly different personalities that get randomized with each clone. So the future of the technology will have to somehow figure out how to get better control over the exact growth habits of the clones' brains. Because if you really wanted perfect copies, the only other way would be to make a lot of clones and then purge the ones that are the least like the original. But that gets dark real quick. But, of course, even a working light bulb could be improved on. And that brings us back to the other way to make clones. Stem cells. The neat thing about stem cells is that they can become any cell, and all cells contain the same code. So theoretically, with the right trigger, even a differentiated cell can be forced back into a stem cell state. The first ones to figure this out were Shinya Yamanaka and Kazutoshi Takahashi of Kyoto University back in 2006. The so-called Yamanaka factors are a group of proteins that, if expressed in a cell, forces it back into a stem cell state. This was revolutionary because up until then, the only way people knew how to get stem cells was to study embryonic tissue. This discovery kicked off a chain of discoveries that are quickly changing the world, even if you didn't know it. For example, stem cells made via this process are now being used in more and more therapies that are treating diseases that we thought were untreatable. And once you have stem cells, you can make any cell you want, like for example, sperm or eggs. If a couple wanted a child, but one of them was incapable of producing either a matching sperm or egg, you could take a skin sample from them, do the conversion, and then use it to create an embryo from both parents. This could be a gay couple, obviously, but just the same, if a heterosexual couple had issues one way or the other, they would benefit from the technology too. For cloning, though, this ability means that if somebody wanted to clone themselves, they don't even need a donor egg anymore. They could make one themselves regardless of if they have ovaries. However, the science is getting to the point that this step might not even be necessary. Something which has emerged in the last few years are what are called stem cell-derived embryo models. You start with stem cells and then split them into a few groups. Each group is treated to kick off a different set of genes that are known to be associated with the first few divisions of an embryo. You then mix a specific proportion of these different cell types into a special cell culture mold that will make all the different cells stick together into a little clump. If you do it right, the blob of cells start doing the embryo thing and growing structures like a heart or other basic organs. The tech is still new, so it's not been used to make a whole animal yet, but it's getting there. Finally, let's talk about the last problem that separates sci-fi cloning from the real thing. Clone tanks. I love a clone tank. Nothing makes something feel more science fiction instantly than a good clone tank. But actually making one is tricky, but probably not for the reasons you think. It's one of those problems where the start bit and the end bit are easy, but there's a piece in the middle that absolutely wrecks you. We can grow embryos in test tubes just fine, and once an embryo has grown to the point that it's got a connected blood supply, we can then take it out and put it into a special culture bag system and keep them alive until birth. But that in-between stage is a bitch. The process of placenta and umbilical cord formation is one that is as coordinated by the mother as it is by the growing embryo. So while new advancements mean that we can keep embryos growing and alive for longer than ever before, getting over this part is where the technology is currently stuck. Recently, scientists managed to keep mouse embryos alive for more than half of their gestation period, using this really cool carousel of glass wombs. But the researchers know that they've come up against the hard bit. So let's finish up by talking about how we, as a bunch of mad scientists on a budget, could make a clone. Well, we love a tech tree on this channel, so let's take a look at ours. As it so happens, our other projects have been actively crossing off many of the pre-work steps to this, so in terms of actual execution, there's a lot less to do. First and foremost, we need a microinjection system to be working. Even if we use an animal that has nice large eggs like a chicken or a fish or a frog, the nucleus itself is still tiny. This means that we need the microneedles and an XYZ control system and a second system to control a very light vacuum for holding onto the eggs. Thankfully, we already have a bunch of really nice microscopes meant for this kind of thing, so we should be able to just mount some new hardware to them. One of the benefits of working with animals that lay eggs is that all we need to do is modify an egg and get it replicating. After that, the egg can do all the growing on its own. This makes the tech tree hilariously short and avoids the need for a clone tank entirely. Nature already made a clone tank, we'll just borrow one. But that does raise an interesting question. Does the species that the egg comes from really matter? Like, if we got salmon eggs, could we grow something other than a salmon in them? Let's say another fish, for example. But if that works, how distantly related can a species be and have the process still work? For some reason, I just can't get the idea of a mouse hatching out of a chicken egg out of my head. Like, it shouldn't work, but I don't know that anybody's actually checked. So if you want to find out and come on this journey with us, then be sure to subscribe so you don't miss it. And if you'd like to show off your mad science flair while supporting our work, check out the amazing items and designs we carry in our store. We've got t-shirts, DNA, bacteria, ancient papyrus, and more. A very normal selection of items for you to peruse. Alternatively, you can become a patron of the channel and get access to our exclusive Discord. And our patrons have been so important in keeping the channel going through the years, so thank you so much. But that's all for now, and we'll see you next time.